Hello, my name is Andrew Roulard, and this is the second part of the lecture on resources for bioinformatics and machine learning. So the next resource we'll talk about is called UniProt, which stands for the Universal Protein Resource. And this is a database that contains all kinds of different information about proteins. You can access this page at the website shown on the right of this slide. And I was specifically going to talk about how you can get protein domain information out of UniProt. This is pretty useful information. A protein domain is a sequence of amino acids that form a particular structure, three-dimensional structure in a protein that has, is known to have a particular function, and it shows up in, often shows up in many different functionally related proteins. So this is useful information for learning about how proteins are related to each other. So on this next slide, you see a data table that I obtained from UniProt. UniProt has a pretty useful interface where you can basically design your own data table. You just make selections for what kind of information you want to be put in this table. It's generated, and then you can download it. So in this case, you see this highlighted entry on the left of the table. This just has a list of protein identifiers. And then what I asked to be included, shown at the right of this table, are different domain annotations from several different other databases that are pulled into UniProt. So these are in the form of identifiers, which you can then convert to actual names for these domains. But what you can do with all this information is then, again, organize it into a matrix format, shown here in this next slide as a heat map, where we have proteins on the left and domains labeling the columns on the top. And we can see now this big picture of how proteins are related to each other based on how they share certain domains. The next resource that I want to talk about is called DrugBank. This is a database of information about drugs and their targets. And by target, we usually are referring to a protein that a drug interacts with to modify its activity. But it doesn't always have to be a protein. The screenshot on the right is again showing you how to access data in the DrugBank database. And so this has information about drugs, such as their chemical composition, molecular structure, different types of pharmacokinetic and dynamic properties, mechanism of action, and then for the targets, which again are generally proteins, there's going to be sequence information, what kind of reactions they participate in, or pathways they participate in, or their cellular localization, and more sorts of annotations like that. So, one of the simplest things that you can do from this data is just make a network connecting drugs to their targets. So this next slide is showing you a data table obtained from DrugBank. And this is in the form of a list, but it's a little bit different from the list we've seen before because for each row we have a protein, which is shown in this highlighted column that's in the center of this table. But then to the right of this table, we've got another column that doesn't just have a single drug, but it actually has sets of drugs. So each row is telling us about a protein, and then all the drugs that are known to interact with that protein. So again, we can parse this information out of this data table and create a matrix, visualized again as a heat map in this next slide, where we have proteins labeling the rows and drugs labeling the columns, and we can see, which proteins are targeted by which drugs, and try to understand where there are opportunities to develop drugs, or how we can use this information to learn about if we know a protein is important in a particular disease we're studying, which drugs can we use, potentially reuse for that disease. The next resource is called ENCODE, which stands for Encyclopedia of DNA Elements. So the ENCODE is a genome mapping project. And so what that means is the aim of the project is to, define, for all these different regions across the entire sequence, across the entire human genome, what is the purpose of every particular region? Is there a gene in this region, or is there a promoter for the gene, or is there a binding site for a transcription factor that's going to regulate the expression of a gene? So that's the example that I'll talk about here, which is transcription factor binding site maps. You can access data and learn more about ENCODE by going to this web page shown in the screenshot on the right. And I just want to mention a related resource called CHIA, which has accumulated this particular type of data, this transcription factor binding site data, from studies in the literature that are not part of the ENCODE project. So the value of this transcription factor binding site data is that we can learn which of these transcription factors are potentially regulating expression of a gene. So in this next slide, we have an example of what some of this ENCODE data looks like. We have, in the first 3 columns of this data table, information about binding site. So this is a genomic location defined by a chromosome, and then coordinates in terms of base pairs or sequence position. And then right next to that we have listed in the column highlighted most to the right, transcription factor. So this is summarizing a whole bunch of these transcription factor binding site mapping experiments. Every row is telling us this transcription factor has been observed to bind in this genomic location. And so what we can do with this data is we can also bring in information about where all the genes are located in the genome and convert all these genomic coordinates to actual genes, and then form a network shown in this next slide where we're connecting genes, which are labeling the rows of this matrix, connected to transcription factors, which are labeling the columns. And so now we've got this network visualized as this heat map, which tells us, a lot of information about how genes are regulated by these transcription factors. The next resource I'll talk about is called GO, or the Gene Ontology. And this is a project that seeks to annotate genes using a structured vocabulary of terms for molecular functions, biological processes, or cellular components in which these genes participate. The screenshot on the right is showing you where you can go to access the data in Go. And there are other, gene annotation resources available. For example, gene reference into function, or GeneRef, is available through the NCBI gene database. And this is a project that, again, is seeking to annotate genes with short statements about, functions that have been documented in the literature that these genes are involved in. This next slide is showing you what the Gene Ontology data set looks like if you were to download it. Again, this is just a list of interactions. We're really just looking, if we just want to get the interactions, we're just looking for 2 columns in this data table. One that tells us genes, gives us gene identifiers, and then another column that gives us, in this case, Go term identifiers. And each of these Go terms is actually has a text description attached to it that we can also look up that is describing a particular function. So we can download this data and parse it again into one of these matrix formats. And now again we see this big picture about how genes are related to each other based on these gene ontology terms that they share. The next resource that we'll talk about is called Reactome, and this is a database of biomolecular pathways. Each pathway consists of a subnetwork of molecules that have been documented to interact to perform some kind of defined cellular function or process. Reactome is one of many pathway databases. Other examples are KEGG, which stands for Kyoto Encyclopedia of Genes and Genomes, and Wiki Pathways. And so these pathway databases are useful for looking for signaling and regulatory networks in which genes are known to participate. This screenshot on the right again shows you where you can go to access information about Reactome and get the data. The data table on the next slide showing you one way to format this pathway information. This format is called a gene set library. Every row defines a set of genes. The first entry in this row is a label for the set of genes. In this case, it's the name of the pathway. And then the rest of the row is listing this set of genes. So again, we can reformat the same information into a matrix where we have genes connected to pathways as shown in this next slide. And again, this is a really nice way where we can see how genes are related to each other based on these pathways that they participate in.